Good afternoon, everyone. My name is Manuel Picon-Ruiz, and I'm actually at the University of Granada, and I'm going to present our last publication. It was published in Cell Metabolism last year, entitled, let me show this, the major pre- and post-menopausal estrogens play opposite roles in obesity-driven mammary inflammation and breast cancer development. So I will start with a brief introduction about this research. Okay. So I will talk a little about breast cancer and also obesity, inflammation, and their relation. Okay. So as probably everybody knows, breast cancer is the most common cancer and the first leading cause of cancer death in women worldwide. You can see here that the incidence of breast cancer is almost 25% of all cancer diagnoses in women, and the mortality accounts for 15% of all cancer death. And breast cancer incidence rises with age, and particularly after menopause. You can see in this graph how the incidence clearly increases with age, and particularly after menopause. So breast cancer incidence is greater in postmenopausal women. There are different molecular subtypes of breast cancer. They are classified based on the expression of hormone receptors, HR. They are estrogens and progesterone receptors, and also the expression of the protein HER2. And the majority of breast cancer diagnoses are hormone receptor positive. But as you can see here, this accounts for all breast cancer hormone receptor positive cases diagnosed in premenopause, in the perimenopause stage, and after menopause. And we can see how incidence of hormone receptor positive breast cancer subtypes increase also after menopause. Okay, this is about breast cancer, some notes about breast cancer that probably everybody knows. I will talk a little about obesity. As everybody should know also, obesity is an increasing problem in the world. Based on data from the World Health Organization, in 2014, 39% of adults aged 18 or older were overweight. That means a body mass index equal or greater to 25 kilograms per meter square. And 13% were obese, so BMI greater or equal to 30 kilograms per meter square. Thus, nearly 2 billion adults worldwide are overweight, and of these, more than 500 million are obese. You can see here, this is not the prevalence of BMI greater or equal to 25, so overweight or obese. In 2014, is not equal distributed in all the countries. But in most developed countries, there is this prevalence is greater to 60%. And also the problem is that this prevalence is increasing over time. You can see here for the United States adults, the trends in overweight, obesity overweight is in this yellow color, and all these pink red colors represent different obesity grades, 30 to 35, 35 to 40, and greater than 40. You can see here how this the prevalence of overweight and obesity are increasing over time. In the data from the 2015, we can see more than 60% of adults in United States with a BMI greater or equal to 25, and of these, almost more even than 50% were obese, when in 1975, less than 15% of adults were obese. And obesity is important for breast cancer and for many different cancer types because it's associated with an increased risk of development, cancer development. In particularly for breast cancer, is only associated with postmenopausal breast cancer in women, and particularly for those that express the hormone receptor, particularly the estrogen receptor. As you can see here in a publication by Renehan et al., you can see that even they found a 10% risk reduction with increased BMI in breast cancer positive for estrogen receptor and progesterone receptor before menopause, but this was clearly correlated an increase in BMI with an increased risk of developing postmenopausal hormone receptor positive breast cancer. And you can see here also in this map different colors that represent the breast cancer cases diagnosed in postmenopausal women that are attributable to obesity. And you can see here for the United States, or Spain, or many other developed countries that these cases account for more than 13% of all the cases diagnosed. In any case, where there are differences in the association between obesity and breast cancer risk, it is clear that obesity is a poor prognostic factor for all kinds of breast cancers at any age, in different subtypes and pre- and postmenopausal cancers. As you can see here, independently of when BMI is diagnosed before or after cancer detection, when you compare obese patient versus normal weight patient, always the overall survival rate is lower for obese women, independently of the diagnosis of the BMI. Okay, so what about obesity and inflammation? It is well known that obesity induces a chronic inflammatory milieu in the adipose tissue. You can see during weight gain, there are changes, important changes in the adipose tissue that produce a change in the composition with infiltration of pro-inflammatory immune cells, change in leptin and adiponectin release, and also the secretion of many important pro-inflammatory cytokines by the obese adipose tissue. We can say that really the adipose tissue changed from an anti-inflammatory profile in the lean adipose tissue to a pro-inflammatory profile in the obese adipose tissue. And obesity is also related with estrogen synthesis, okay? After menopause, everybody knows that ovaries stop producing estrogens and estradiol decreases markedly, becoming strong the major serum estrogen. As you can see here, how the E2, so estradiol, E1 is strong, so estradiol-strong ratio is changed from greater than 1 to lower than 1 after menopause. No? So in the premenopause phase, estradiol is the major estrogen in the serum, and after menopause is strong, the one that becomes the major serum estrogen. Also, importantly, obesity and serum estrogen levels are associated after menopause. You can see with the increase in BMI how strong and also estradiol, but mainly strong levels increase. You can see here also how strong values measured in picograms per milliliter are greater than for estradiol after menopause. No? So BMI correlates with an increase mainly in strong levels. Okay, and obesity and higher strong levels. You can see here. This is comparing the highest quartile again compared to the other, the lower strong levels, and also for BMI and estradiol. So we can see that highest strong levels are correlated with increased risk of ER positive compared to ER negative cancer subtype, also with progesterone receptor positive, and also with ER positive, PR positive cancer subtypes, when estradiol is not. So high strong levels correlate with greater risk to develop hormone receptor positive breast cancer, similarly to also the increase in BMI. So importantly, the obese adipose tissue is directly involved in estrogen synthesis. As probably you know, aromatase is the main enzyme involved in estrogen synthesis. Aromatase converts androstenedione into estrone and testosterone into estradiol. Right? And aromatase is highly expressed in the fat tissue, and after menopause, when ovaries stop producing estradiol, estrogens are mainly synthesized in peripheral tissue as the adipose tissue. No? And importantly, not only the adipose tissue became one of the main sources of estrogens after menopause and in the form of estrone, but also aromatase expression is increased in the obese adipose tissue. This is shown before in the figure before when we compared the anti-inflammatory versus the pro-inflammatory profile in lean and obese adipose tissue, but also the NF-kappa B pathway is one of the most important pathways altered in the obese adipose tissue that is associated with the inflammatory milieu. Also is involved in increasing expression and activity of aromatase. Finally, what is the consequence is an increase in estrogen synthesis. And remember that these estrogens are mainly in the form of estrone. And the NF-kappa B pathway, we are showing that is activated in the obese adipose tissue, is also involved in cancer development and progression. This is very simple diagram where we can see how NF-kappa B pathways is related not only with inflammation, but also directly related to cancer. And also pro-inflammatory mediators also are known to be involved in cancer development and cancer progression. In particular, it's known that the NF-kappa B pathway have different roles in cancer cells, including survival, proliferation, invasion, angiogenesis, and metastasis. By last, I want to introduce our hypothesis. So the estrogens, when we wanted to analyze this, to compare if estradiol and estrone had different biological function, at that time we knew that estradiol was very well known to decrease inflammation through the inhibition of the NF-kappa B pathway. It has been studied in many different cell types and have been identified different mechanisms in which estradiol bound to estrogen receptor inhibit the NF-kappa B pathway by inhibiting the translocation to the transcription NF-kappa B transcription factor A, CREL, REL-B, etc., to the nucleus, and also even in the nucleus by different mechanisms in making that the target gene from the NF-kappa pathway are not transcribed. Okay, so but we didn't find in the literature what is the role of estrone on NF-kappa and inflammation. During the publication of our previous paper, this paper that I am showing here, published in Cancer Research, where we analyzed the interaction between adipocytes and brain cancer cells, we really used a model that finally we call it pre-adipocytes, because where adipocytes differentiated from mesenchymal stem cells. So remember, one of the reviews asked if this cell type was really compared, has a similar function that adipocytes, and we make a comparison between mesenchymal stem cells and differentiated to pre-adipocytes and with adipocytes, measure adipocytes directly isolated from the adipose tissue, from the abdominal and also breast parts. And we found, interestingly, that co-culture with measured adipocytes directly isolated from the fat tissue, have a greater effect on ER positive. These are in red. Okay, we used 4 different ER negative breast cancer cell lines and 3 different ER positive breast cancer cell lines. You can see here for blue for the ER negative, red for the ER positive, and we assigned by qPCR different pro-inflammatory cytokines. We can see here CCL2, IL6, and IL8. And in all cases, adipocyte co-culture induced a greater upregulation of this cytokine in ER-positive breast cancer cells. We also found that that different embryonic stem cell transcription factors, very important in pluripotentia and cancer stem cell biology, like SOX2, NANO, and OCT4, were also greater upregulated in this ER-positive than ER-negative breast cancer. So we started to think that it has estrogen component, no? This has the difference between the effect in one's breast cancer subtype depend on the estrogen. But we already knew, remember, that estradiol is well recognized to be an inhibitor of this pathway that releases the pro-inflammatory cytokine, not the NF-kappa B. Interestingly, we found that in our co-culture system, you can see here the control, how you can detect clearly both estradiol and estrone, but in our co-culture system, this means plus adipocytes, so breast cancer, ER-positive breast cancer cell co-cultured with adipocytes. In the supernatant, we only have mostly estrone. So, based on all this, we make the following hypothesis. No? We hypothesize that estrone drives ER-positive breast cancer, both development and progression, by promoting obesity-mediated NF-kappa B activation. So we already know that estradiol inhibits this pathway, but we made the hypothesis that maybe estrone was activating this pathway. No? And in addition, we also hypothesized that enzymes that convert estradiol into estrone, are oncogenic because they increase intracellular estrone levels. These enzymes are mainly a family known as HSD17B. There are several 15 members, 15 isoforms, and many of them produce the conversion of estradiol into estrone, and also some of them estrone into estradiol. So they are affecting the ratio estradiol, not only directly with estradiol and estrone, but also in the precursors, androstenedione to testosterone, etc. Okay, so I will present now our results that were published in this paper, no? In the Cell Metabolism paper last year. I'm going to show only the most important for not being too late, okay? So the first thing that we saw is that inflammatory cytokines increase with obesity, menopause, and cancer. You can see we tested by qPCR these different pro-inflammatory cytokines in non-obese versus obese fat tissue. This is adipocytes isolated directly from breast fat tissue from patients, okay? And you can see in all cases that obese adipose tissue isolated from breast fat have greater expression of these pro-inflammatory cytokines. We also compared these values between breast fat from premenopause compared to postmenopause women. And you can see not significant difference for CCL2, but a clear significant difference for IL-8, indicating even when this was not significant, it was showing a threshold for increased cytokine, pro-inflammatory cytokine production in adipose tissues from obese postmenopausal women compared to obese premenopausal women. And also, importantly, these assays were performed with healthy patients. Here we compared also from 3 different patients with that we obtained breast fat from the breast affected by cancer and the unaffected breast, the contralateral unaffected breast, and we compared the levels of these pro-inflammatory cytokines in the normal breast compared to the cancer breast. You can see that the fat tissue surrounding the cancer has also showed increased expression of these pro-inflammatory cytokines. By last, we'll present here also the data how our estrogen receptor positive breast cancer cells increased upregulate the expression of these cytokines when co-cultured with adipocytes obtained from pre-menopausal breast fat and from postmenopausal breast fat. You can see the co-culture with adipocytes from postmenopausal women also had a greater effect to upregulate CCL2, IL-6, and IL-8 in our ER-positive breast cancer cells. Then we also wanted to determine if this effect, the effect of upregulating cytokine, pro-inflammatory cytokine expression in the ER-positive breast cancer cell by adipocyte was dependent on aromatase activity, so on estrogens and NF-kappa B. So we use an aromatase inhibitor, is for letrozole, okay? And we saw that when we added letrozole to our co-culture system, the upregulation of the cytokine, pro-inflammatory cytokines in the ER-positive breast cancer cell were clearly decreased, okay? Also, similar result we observed with an NF-kappa B inhibitor. When we added an NF-kappa B inhibitor to our co-culture system, also abolished partially the upregulation of the pro-inflammatory cytokines in the ER-positive breast cancer cells. Based on this, we performed an RNA-seq study to compare if both estrogens had a similar biological function in global gene response, okay? And as you can see here, when mostly of the genes upregulated and downregulated by both estrogens were similar, we found several genes that have a different activation or downregulation by estradiol and estrone, okay? Importantly, you can see here some genes that were activated, upregulated by estradiol and downregulated by estrone, and the opposite, okay? Downregulated by estradiol and upregulated by estrone. And from these up genes regulated by estrone, we found that they were they were related to the TNF-alpha signaling via NF-kappa B. TNF-alpha is a known activator of the NF-kappa B pathway. So these activated genes by estrone, but not estradiol, were associated to NF-kappa B activation. We also wanted to, we also studied by RNA-seq the effect of estrone and estradiol addition when ER-positive breast cancer cells were treated with TNF-alpha. We activated the NF-kappa B pathway by treatment with TNF-alpha alone or in combination with estrone and estradiol. And as you can see here, many genes activated by TNF-alpha were more activated when we added estrone, while were downregulated with estradiol. This result were also predicted based on the previous literature about the role of estradiol in NF-kappa B pathway inhibition. But this was very exciting data. No? We also found that these genes more upregulated by strong addition were involved in very important pathways related to cancer. Okay, you can see mTOR signaling, insulin resistance, pathways in cancer, adipocytokine signaling, thyroid hormone signaling, and cell cycle, for example. We also wanted to see effects in vivo. So we performed a study with a mouse model that expressed the luciferase after a NF-kappa B binding site. So that means that they produce luciferase when NF-kappa B is activated. And we made a study using two different diets. A low-fat diet, so mean that the mice were non-obese, and also mice fed with a high-fat diet, okay? That is the yellow line. As you can see, and we expected, we measured in the abdominal region, but this data also is only from the mammary glands, okay? It was measured in the mammary gland, were the similar results. But here, showing the data for the mammary gland, we see that high-fat diet, so induced obesity in mice, produced a greater luciferase expression. That means an activation of the NF-kappa B path in vivo, okay? Importantly, we saw that when we added a strong, so we introduced pellet implants in the mice with a strong or estradiol. So you can see the red line, how the role for the relation between luciferase activity, that means NF-kappa B activation, and weight was greater, so by strong than for the control, and also by the estradiol. So estradiol even decreased a little the effect of NF-kappa B activation by obesity. We compared by weight, making a comparison that like mice with weight lower than 40 grams were non-obese, and greater than 40 were obese, okay? We compared these values, and you can see how the luciferase activity, so that means the NF-kappa B activation was greater in the mammary gland of obese mice when we did an implant, so in the placebo group, in the control group, and was greater for the strong and lower for the estradiol. Okay, you can see that even estradiol, obese and non-obese mice did not differ significantly in the NF-kappa B activation. Okay, so by last, we wanted to analyze also I was mentioning before in my hypothesis that we also thought that some HSD17B enzymes that convert estradiol into estrone may have an oncogenic role in breast cancer. So here, I'm presenting the data comparing the expression of one of these enzymes, particularly the HSD17B14 isoform that produce estrone from estradiol. We compared the expression in adipocytes surrounding the cancer and from adipocytes isolated from non-tumor bearing breast. Okay, so non-cancer breast and cancer breast, and you can see how HSD17B14 expression was higher in adipocytes surrounding the breast cancer tumor. Okay, and we also analyzed public data from patients, which found that high intratumoral expression of HSD17B14 in ER-positive breast cancer patients correlate with lower overall survival. So the red line is representing the highest HSD17B14 expression values in the tumor. Okay, you can see a significant difference in the decreased overall survival by high HSD17B14 expression. Then we performed an in vitro experiment where we overexpressed this enzyme in ER-positive breast cancer cells. We used two models here, MCF7 and D47D breast cancer cells. Both are ER-positive breast cancer cell lines, and we, as we expected, overexpression of HSD17B14 increased estrone levels. Okay. And decreases estradiol, of course, okay? Because I want to remember again that HSD17B14 convert estradiol into estrone. For that, we found greater levels of estrone and lower levels of estradiol in our overexpressed cells. Interestingly, also, not only it changed the values of intracellular estrogen, but also these overexpressing cells showed increased values for the pro-inflammatory cytokines I'm showing here, analyzed by cytokine array, and also I'm showing here the values for the ESTF markers, no? The SOX2, OX4, nano, and CIMIC. They are embryonic stem cell transcription factor related to pluripotency and cancer stem cell biology. So they were increased in the HSD17B14 overexpressing cells. And also, in functional assay, we found that the treatment with strong, but not estradiol, or the overexpression of this HSD17B14 enzymes, that means the increase in intracellular strong levels induced a greater ability to form mammospheres by these ER-positive breast cancer cell lines, also indicating, correlating with this data, that they acquired a greater cancer stem cell feature. Okay, we also compared by RNA-seq the global expression profiling in MCF7 cells wild type and overexpressing HSD17B14. We found important upregulation of pathways involved in cancer, CARAT, WNT, angiogenesis, HESCO, and also upregulation of basal-like phenotype, also metaplastic perisubductal, and this, okay, down luminal mature, so they were more undifferentiated, we can say. So we used this cell model to perform an in vivo assay for tumor growth. You can see here how the injection of MCF7 cells overexpressing this HSD17B14 enzyme is a more faster tumor growth in vivo, okay? And also, we compared strong versus estradiol, and we saw that treatment with exogenous estrone by pellet plant in the mice also induced the growth of the tumor in the MCF7 wild type, okay? So, estrone induced a greater tumor growth and also overexpression of HSD, so conversion of estradiol into estrone intracellularly in the tumor also induced a faster growth of the tumor, okay? As you can see here, in these groups that has the high and the faster tumor growth rate, also has the greatest estrone levels, intratumoral estrone levels, okay, and the lowest estradiol levels, okay. So that means that highest estrone, faster tumor growth. And similarly, we found not only the tumor growth was increased by estrone, both by exogenous exposure, by pellet implant, or by intracellular conversion by HSD17B14 overexpression, but also in a limiting dilution assay, we found also that tumor initiation stem cell frequency was greater in the groups that were treated with estrone or were implanted with MCF7 HSD17B14 overexpressing cell, independently of the use of estrone or estradiol supplement. Okay, so I will finish here with the results. I will make some conclusions, and we'll talk about the clinical relevance that we found from this result. So the conclusion from this work are that menopause, obesity, and cancer increase pro-inflammatory cytokines in human breast fat. Also, adipocyte-driven inflammation on ER-positive breast cancer cells is mediated by strong synthesis through aromatase activity and NF-kappa B pathway activation. Strong stimulates and estradiol relieves obesity-mediated inflammation through NF-kappa pathway activation in vitro and in vivo. Importantly also, HST17B14 upregulation increases intracellular strong to drive inflammation and ER-positive breast cancer stem cell expansion. And high intratumoral strong levels correlate with faster ER-positive tumor growth and greater tumor initiation ability of ER-positive breast cancer cells in vivo. And I will finish with this clinical relevance that our results we think they have for the ER-positive breast cancer cell biology. So in the clinical, we think that this result demonstrates for the first time a differential role of estrone and estradiol in regulating obesity-mediated NF-kappa B activation and may serve to develop safer hormone replacement therapies for postmenopausal women. This work also revealed that the potential value of enzymes that convert estradiol into estrone, particularly HSG17B14, that it was the one that we tested here, as new marker for the early diagnostic and prognosis of patient outcomes in ER-positive breast cancer. Moreover, these results identify enzymes that convert estradiol into estrone, particularly, again, HSD17B14, as potentially better therapeutic targets for ER-positive breast cancer treatment. I want to finish giving thanks the Joyce Slingerland's lab. Almost all the work was performed at the University of Miami, thanks to the grant funding my work. It was the Susan G. Komen Foundation, and also the last part of the project was performed at the University of Granada in Dr. Mark Charles' lab, thanks to the grant funded by the Marie Curie Actions. So thank you very much, and I'm giving here again the reference of the publication if someone is interested, and also my email address is anyone who want to contact me.